Hello everyone, I'm excited to welcome you all to this plenary session on priority pathogens, particularly focusing on interoperability and continuing that theme that we started talking about this morning in the great introductions and the panel discussion. And this is really focusing on that data standards and contextual data frameworks to enable connected, shareable, and reusable genomic surveillance across regions. We're going to have 6 excellent speakers, some of which you've already met. And then what we're going to do is we're going to combine the question and answer towards the end of the session. So if you think of questions during individual speakers, please make a note and make sure to ask it when the last 20 minutes we have dedicated for Q&A. So the first speaker today is Emma Griffiths, who again you've all just met, who's going to talk about a lot of the work we've done within the PHA4GE Data Structures Working Group on these data standards for genomic surveillance. Emma Griffiths.
Hello, hello, hello. Welcome back. As if you hadn't heard enough from me already this morning, you're now going to hear a little bit more about who we are and what we do in the Data Structures Working Group. So as Finn mentioned, my name is Emma Griffiths. In addition to being a co-chair of the Data Structures Working Group, I'm also a researcher at Fraser University. We sort of talked about this earlier this morning. The focus of our research is developing methodology for data standards development, and we apply this methodology to creating data standards for Canada that are also used elsewhere. But in the context of PHA4GE, we also use that same methodology to build consistent, interoperable, consensus-based data standards. So as Finn mentioned, the purpose of this talk is to give you a little bit more information about our activities and our ongoing projects. I am the mouthpiece today. I have the luxury of standing up and being able to share our activities with you, but the work that I'm about to present is really the work of many members in our working group, but also many community partners. And in fact, data structures, just put your hand up. Put your hand up really high so everybody can see who you are. Awesome. People, if you're sitting next to a data structure, give them a high five. Awesome, awesome. And I promise you that's the interactive section of our session this afternoon. That's over with now. People have been telling me to make a joke that the reason why I'm so hoarse is because I talk about data standards so much, or that data standards aren't sexy and I'm trying to sex it up with this voice. But really, I just have a cold and I sound like Marge Simpson, and there's nothing wrong with that. So now we're going to move on. Data Structures Working Group, who are we and what do we do? Our scope is about developing, documenting, and implementing best practices for structuring data values and the relationships between them. It's about improving data interoperability, data integration, and data management in and across different public health settings all around the world. So how do we go about building data standards? The really first critical step is interacting with the community in order to identify a challenge that could potentially be solved by a data specification. So whether it's because of the experiences and the challenges with members within the working group, or whether members of the community corner us at a conference or send us an email, that integration and that collaboration with the community is really the most important step. Next, if we identify a challenge and we think we can solve it with a data standard, we go about building that standard with a particular methodology that I'm going to highlight in just a moment. And critical to the success of data standards is their implementation. A standard is only as good as its implementation, so once we develop a standard, we must integrate it and put it into practice in different kinds of tooling, and we also create a whole wide range of support materials that help put that data standard into practice. Things like curation SOPs that highlight different practical, ethical, and privacy considerations, different reference guides that define the fields and terms, different submission protocols, etc. After we create the package, the next critical step is testing, and this is where you all come in. Testing, making sure that it works in real live settings, real world settings. We gather feedback from the community and we do our best to respond to that feedback. And finally, dissemination of data standard packages is really crucial. So we publish, we release everything, version-controlled versions of our standards on GitHub. We come to lovely conferences like this and talk until we're hoarse about why they're important. So there are many different types of standards we are currently developing and that we hope to develop in the near future. One of which we're going to talk a little bit more about, and that these are contextual data specifications. These are for the metadata that accompany different individual sequences. But there are also different kinds of standards. Standards for derived or aggregated data, and we sort of alluded to those earlier this morning, that can be used for reports, different kinds of presentations to policy and decision makers. There are different kinds of standards that help to structure information within systems, and there are standards that can be used to help streamline the flow of information within pipelines. There are lots of ways to participate in the development of data standards. So, for example, in the Data Structures Working Group, we have folks that participate in our scoping and needs assessments. So whether you are actually conducting the need assessment because you're a member of PHA4GE, or whether you are the stakeholder that we are engaging, all very important roles. There's also technical development, so knowing the principles and practices, the methodology, the frameworks used to develop standards, that's something that there are different roles for that in the working group as well. As I've already mentioned, testing and feedback, that's very critical. Implementation, people who try out the standards and say, yes, this is actually this is working for us, it's benefiting flow and data quality, and they actually implement it in routine practices. That we love to hear about case studies and collect that, because that helps to convince others to use the standards as well and shows other people models for how they can be implemented. Knowledge mobilization. So if you sit in on a meeting and then you take back what you learn to your home organization, that is very, very important as well. And consensus generation. So whether you are implementing, whether you're in a meeting and you're saying, yes, I agree to what you're doing, whether you come in when the paper's being written, you read it over and you agree, getting your name on that paper to show that support, these are all mechanisms for demonstrating consensus. So as part of our standards development methodology, we have a modular interoperable framework that we use for all of our different standards. When I say that it's modular, what I mean is all the fields, terms, and formats are grouped together thematically into different modules. Those modules can be for things like database identifiers, sample collection and processing, sequencing, diagnostic testing, etc. All of the fields and terms that we populate the modules with are sourced from ontologies. I don't really have time to tell you what those are, except that they're international semantic resources that are really great sources for international vocabulary. When I say that the framework is interoperable, what I mean is that each of the modules can act independently. It's almost like a plug-and-play system. So you can develop a new standard for pretty much any pathogen or any target just simply by mixing and matching the different modules and also customizing what's inside those modules. As I mentioned, all of our standards are supported by tooling and reference material. They're all maintained. We have a commitment at PHA4GE to maintain our standards. They're version controlled. You know, the standards are not one and done. As data needs change and evolve over time, the data standards have to keep up with those needs. And we have mechanisms, and we really encourage participation and communication between data standards developers and users. Just to give you a little bit of an idea, what the modularity of the standards look like, I've done a little diagram. On the top level in blue there, those are the modules of an MPOC specification that has been developed based on this methodology by CIDGO, by my lab. And on below, in red, are the modules for the PHA4GE wastewater surveillance specification. And you can see through the kind of crosshatch vertical lines where the modules align in both standards. And what this suggests is that if you are a public health agency and you are surveilling, or if there are different labs surveying MPOCs using different strategies, one using clinical data, another using wastewater or other environmental data, the data generated by those two different strategies is much more integratable using compatible interoperable standards than if you were using two completely different standards or no standards at all. So I alluded to a formalized methodology earlier on. We've sort of boiled down that methodology into 10 simple steps. They're not really as simple as I'm making them out to be, but for the purposes of this presentation, you can see them here. I'm not going to go through each and every single one, but I just wanted to emphasize that the 10-step approach really emphasizes building interoperability, co-creation with the community, operationalization, so putting standards into practice, and real-world implementation. Also, yes, I know that there's that cartoon that describes the proliferation of standards. People have sent that to me 1 or 2 million times, so no need to send it to me again, if that's what you had in mind. So the methodology and the framework has been used to create a growing library of interoperable specifications. For example, I have a few examples here with regards to contextual data standards. These are necessary because different labs collect and store data in different ways. They're asking different questions, they're using different concepts, they're collecting information at different levels of granularity. And that creates real challenges for data integration and data exchange, and using that data for different kinds of analyses. So our contextual data standards are meant to harmonize sample metadata as well as different kinds of epidemiological, clinical, environmental data, lab results, and critically capture methods. And I think there was a great question this morning about that. Like I mentioned, our contextual data standards are implemented in a data collection, harmonization, validation, and transformation tool called the Data Harmonizer that Charlie gave a little cheeky shout out to this morning. And we have many global implementations for our contextual data standards. So the two I just wanted to briefly mention were our SARS-CoV-2 standard, which was developed during the pandemic. This was our sort of flagship data standard. It's implemented by many labs all over the world. We also have just put out a wastewater surveillance data standard. This is because wastewater is an increasingly used tool in public health to understand the prevalence and diversity of pathogens in different settings. Again, there's a variety of sequencing techniques that are being used to ask questions of wastewater, and hopefully that standard will help harmonize all of that information. So we have publications, and again, also our standards are available on GitHub if you want to take a look. And I've just put the links there. We've also built standards to help harmonize inputs and outputs of different processes. So after this, well, I guess really the first activity or the first challenge that was brought to us was the idea of being able, as a public health lab, being able to evaluate different AMR detection tools for clinical and public health use. And that can be challenging because different AMR detection tools use different algorithms, they have different scopes, they use different reference databases, and so they generate different outputs. And that can be hard to compare for public health labs in order to evaluate which tool is best. So we have a tool called Harmonization that we created in order to help enable comparisons of different tools across different labs and different settings, and to enable exchange of AMR data. So Harmonization basically harmonizes the inputs and outputs of, I think, 18 different tools. And the outputs that are most useful for public health use have been prioritized, and we've harmonized those. It's been tested and implemented internationally, including by some organizations like PHA and Lulamba. And the beauty of harmonization is that it also produces a really nice summary report as well. So that's available. If you want to learn more about how it was created, how it was tested, how it's useful, we have a publication there as well. Also, we have some specifications that are meant to help data sharing. So our QC specification tag is a set of attributes that help people tag their data sets with information about quality control. So oftentimes public health labs only share data if it meets a really high stringent level of quality, but data that may not meet those stringent conditions and may be of slightly less quality are still useful for a lot of things. You can see on the slide here things like training and testing tools, and as well as other different types of analyses. And so in order to better enable labs to share data sets of different quality, we've put together this small group of attributes that allow labs to tag their data sets with how something was QC'd, what the ultimate result was, and any kind of known issues. And that also helps people identify data sets with known issues if you're wanting to do any of this sort of tool development, tool optimization training as well. We also have the INSDC compatible pathogen data object model. Essentially, this is just a set of rules and attributes for sharing data to the INSDC. A lot of this was already in place, but it really helps to advocate for the use of biosamples, and it provides additional attributes. So if you're wanting to describe your samples using additional attributes that are not currently in different mix packages, those are available. It also encourages people to add more metadata for the sequence data as well, and it basically just describes where those different types of metadata should go. So I wanted to quickly allude to our ongoing and upcoming projects. So in terms of contextual data, we're currently building a minimal contextual data specification, and this is because usually when people embark on a new sequencing initiative, or surveillance initiative, they have to have a big argument about what should the metadata be. And so to sort of reduce those arguments, we've created a minimal specification that is in all of our different contextual data standards. So just in the process of working that out. We're also building a highly pathogenic avian influenza data specification to be able to track HPAI. This is also going to serve really as a model for segmented viruses. You know, we've worked with SARS-CoV-2 in the past, but this is going to add on new methodological attributes for segmented viruses. We're also building a malaria data specification. And we've already talked a little bit about that. We're building that in collaboration with the Africa CDC. And this is also going to serve as a model for vector-borne diseases in general. So again, reuse, reuse, reuse. In terms of additional standards for inputs and outputs, we are currently creating a specification for genotyping. So creating a set of attributes that will allow people to search and filter for different methods and different genotypes. We will be, I'm describing this very quickly right now, but if you're interested in learning more, we're going to talk about it more at our data structures working group breakout session tomorrow. So come and you can give us some feedback on this specification. CONVAST is all about transforming native antimicrobial phenotypic testing data into INSDC submission-ready formats, so you don't have to go and get a printout or whatever and then type it into your AST template. This is going to automate that for you. We also have a project about harmonizing primer schemes. Really what that's about is providing a set of attributes to characterize a primer scheme. There's some tooling to be able to combine that attribute information with bed files, and we have a growing library of characterized primer sequences as well at PHA4GE. You know, when we showcase our contextual data standards at conferences, we often get people coming up to us afterwards saying like, oh, that's really great that you have these contextual data standards, but do you have anything for pipelines? And we're like, not really so much yet. But that is a project that is underway. We're currently assessing and scoping which processes we should be working on next. So we will be making something similar to what we did for our harmonization. We're looking at being able to prescribe standardized formats for QC metrics. We're currently sort of canvassing the crowd about what kinds of metrics are frequently used for particular pathogens in different circumstances. So that's coming soon as well. We have another standard that we're working on that we call What is a Sample? And that's really about standardizing the way we talk about all of the different entities and the complexity of these entities from sample to sequence. So you know, if you have a sample, oftentimes within those samples there can be many different pathogens. Each of those pathogens can have different isolates. Those different isolates can be characterized in different ways, and each of those different isolates may produce different sequences. You can also have different replicates, controls, synthetic data, and so not having a common language to be able to describe all of those entities can really create confusion. And so we're currently working on nomenclature for that. In terms of helping to mobilize data from local to global sources, I think this was mentioned this morning. We are currently working with IPSN on creating a data standards repository so that not just phage standards, but standards that are created in a compatible way in the community. This will provide a one-stop shop for people to be able to find what standards already exist, so you don't have to, proliferate standards like in that comic. And finally, we are really proud to be working with the INSDC. We are currently working with them to create new phage wastewater surveillance biosamples. The one that I have in the screen here, this went live at NCBI a couple of weeks ago, and as Peter mentioned, we're currently working on a separate new phage wastewater biosample package at ENA as well. I'm almost out of time, and that is good because this is my last slide. So in summary, the Data Structures Working Group tries to be as responsive to community needs as possible. We really emphasize co-creation with members of the community, and implementation of standards is really key for demonstrating utility and uptake. The formalized standards methodology that we use at PHA4GE is a really powerful mechanism for creating consistency, interoperability, and as well within that methodology, that's a great way for implementing different kinds of best practices. We have, as you've seen, we have a lot of projects on the go, and we need a lot of help because all of them are only partially done. And so if anybody's interested in participating, I put a variety of ways to contact us in the slide. We would really love to have you. And again, if you want to learn more, come and see us at our breakout session tomorrow. So with that, I just want to deeply, deeply thank our funders, particularly the Gates Foundation, who have been incredibly supportive of developing data standards. All of us are volunteers, so all of us are supported by our home organizations and the grants that go with those. So very much thanks to all of our individual funding agencies, thanks to all of our PHA4GE Data Structures Working Group members, and to all of the partners that make all of this work possible. So thank you very much to the global pathogen genomics community, and thank you for listening.
At the risk of sounding a bit self-congratulatory, thank you for that excellent summary of the Data Structures Working Group work from Emma. So again, remember, if you've got any questions, keep it in your mind, make a note, and ask it at the end of the session. So next we have Charlie Barclay, who's going to talk a bit more about some of that wastewater standards that Emma alluded to, the trials, tribulations, and some of the successes. Charlie?
Thank you for having me, and I promise after this is the last you'll see of my face today. I'm sure you're bored of my face and of my voice. I know that I am. So I'm Charlie Barclay. I come to you from Fraser University, the Centre for Infectious Disease, Genomics and One Health lab with the lovely Dr. Griffiths over there. And today I'm going to be talking to you specifically about one of the projects Emma's already teed up for me nicely, which is that wastewater surveillance data standard project. So I don't think it's going to be a surprise to anyone here when I say that we've seen quite a large uptake in genomic techniques for characterization of infectious disease. But what I would like to highlight is we saw some really great analysis done earlier today, different phylogenetic techniques, and that is only feasible and only doable when we have good quality contextual data. And a lot of our analysis is actually limited by the quality of our contextual data. Now, I know there's a number of bioinformaticians in the room, and we like to use that analogy of assembling a sequence is like trying to put the pieces of a puzzle together. And I think I'd like to take that one step further by asking you to think about the sequence without the contextual data is like having those pieces of the puzzle with no picture in the box. Not to say that you can't do it, but it really is difficult to understand what the picture is saying and to be able to interpret that information. And so I really want to hammer home the importance of contextual data. There we go. So I'm sure none of you are surprised when I tell you that what I think is one of the solutions is data standards. And I'm going to whiz through this slide because Emma's already shown you this for our 10-step process. But yeah, our solution has been to build data standards that bring both that structure and interoperability to this information. And so with this collaboration with SIGGO and PHA4GE, we've developed this sort of 10-step process in order to help us address that messy contextual data challenge, to support that robust analysis that's really important to the analysis and the public health decisions that we make. So within that, Emma's spoken a lot about the modular architecture that we have. And one of the key pieces of this is that there is flexibility through that modularity. So we use standards that have a consistent core, and we've adapted that in order to be able to evolve as the needs and the use and the pathogens that we are supporting and that we are studying evolve. These modules can be combined and they can be tailored to different surveillance needs, different use cases, and across the global need. It's built on a pathogen-agnostic core, which contains some of the really key pieces of information, but using those ontology-based terms, which help to save us well when it comes to the interoperability and mapping and exchange formats that we talk about later. So as I said, using that consistent core and having those interoperable ontological-based modules is really useful for when we're adding on additional fields as needs grow, and especially when we're looking at additionally complex information. And wastewater surveillance really brings that added layer of complexity. So instead of just thinking about individual patients, we're having to think about sort of community-led, so those composite samples that are affected not just by the health of the patient, but also by additional factors like rainfall, the wastewater systems that pertain into them, flow rates, and things like that. So there are multiple different additional parameters that we need to consider and that are involved. Now, in addition to that, we've also got additional stakeholders that come with this as well, with the clinicians and the public health agencies. We've now also got treatment plants and water systems that we have to consider. So there's additional domains of knowledge that we have to work with when we're incorporating and designing these data standards. So this is exactly where a modular and interoperable approach can be really useful, because we can use that core that we've already got, but build upon it to be able to help with these additional parameters and scenarios that we find ourselves in when we're talking about wastewater. So we at PHA4GE were designed with coming up with a wastewater contextual data. And we spoke about that 10-step process earlier, and the first two key points of that 10-step process are scoping it. So really identifying what those aims are going to be for this wastewater specification. So we wanted to try and keep something that was pathogen and sample agnostic, while maintaining that interoperability element with our existing PHA4GE standards. That was a really key part of the process to us. And I have to shout out Gates Foundation, I know Emma's already done that, for funding us, and we really appreciate your efforts for that. We also have incredible collaborators at Public Health Agency Canada, which we also are very grateful for. So at that needs assessment stage, that's when we identified some of the challenges, which was the diversity of the systems, the different environmental sites, the different collection methods and techniques, as well as the variable sampling strategies and sequencing strategies. So what was the result? Well, the result was this. So we have the PHA4GE wastewater contextual data standard, and you can see that we've hosted it on GitHub just behind me here. So this standard is compatible with our existing PHA4GE standard, our SARS-CoV-2, and our minimal pathogen agnostic standard. It is a repository that contains a list of fields and terms in its purest essence. They are all ontology-based terms, modular, and most importantly, they've got clear definitions, URLs, and guidance that go with them. Now, we actually designed this standard to have 3 use case templates. So we looked at SARS-CoV-2, antimicrobial resistance, and pathogen agnostic. And part of the reason we did this was to lower that barrier to uptake. So we've got 3 use case templates in there. However, of those 3 use case templates, we still have these 6 core ontology-based modules that you can see behind me. So that database identifiers, sample collection and processing, sequence info, bioinformatics QC, and the pathogen diagnostic testing are all core and core with the existing standards that we have at PHA4GE as well. Interestingly, the develop of this specification enabled us to come up with a new module, which is environmental conditions and measurements. And that's because we have that additional information that we need to capture, such as rainfall, water flow, as well as the potential sites that the water comes from and the additional sites surrounding that might influence those samples. So how do we lower those barriers of uptake of a standard? It's all great having another standard out there in a GitHub repository and say, please use this is amazing. But it's really important to encourage update, that it's clear, that there's clear guidance. So we develop supporting documentation, standard operating procedures, and worked examples. I think a picture says 1,000 words, but Emma always says a worked example is worth 10 pages of a manuscript. And importantly, we've operationalized it through tooling. So the tool has automated export functions. It's the one that I shouted out earlier when I was off on stage, which is the data harmonizer. It is a spreadsheet-style validation tool, so it's pretty familiar to the kind of Excel, Google Sheets that you might be familiar with, but it has additional validation functions, and that helps to really bring the power and the strength of the data standard. So this step made the standard practical, not just theoretical, and prepared it for real-world testing through the PHA4GE subgrant program. So we've got a standard, we've implemented it, we've got it in tooling, what next? Well, to ensure that the specification actually works in practice, we ran extensive subgrant testing, and some of the funding from the Gates Foundation enabled us to do that. So we had partners from a number of different organizations, both public health labs, academic labs, some industry and international collaborators, which meant that we were able to test this in 16 countries and with 2 case studies that are reported in the paper. So these pilots really assessed the usability aspect, so whether the fields and guidance were sort of intuitive, the interoperability, so how well the data mapped to repositories, as well as the clarity and completeness. And actually, it was the feedback from these subgrants that directly informed revisions before we did our sort of official release last year. So yeah, this phase was really essential before scaling up to this release stage. Now, one of the other barriers to uptake for any kind of data standard is how does it get into downstream repositories? A lot of us, when we're applying for grants, one of the aspects is that we have to share our data openly. So there's no use in me telling you to use my data standards if you've then got to edit it and change it again in order to be able to get it into your repository. So we've mapped the major repository standards for you, and we've automated that export in our tooling, the data harmonizer. So with one button, it's in a submission-ready format. We've mapped to NCBI, both the wastewater and the biosample, as well as the SRA packages, ENA, sludge and sewage packages, as well as the phase, ODM, and GISAID. Actually, I'll just go back there. Just to say, as Emma already alluded to, one of the incredible things is that we've also been working with each of these organizations in order to develop PHA4GE-specific attribute packages, and the NCBI PHA4GE wastewater package is now live, and we are working with the lovely Peter at ENA to work on that one too. So, future directions, expanding interoperability. So looking ahead, this work really does inform the foundation for a program called iMicroSeq in Canada. So this is a national portal that's being developed for microbial water-based genomics, and the PHA4GE wastewater standard is actually being used as the sort of foundation and the building blocks to develop this new water microbial-based standard that will be the skeleton and the backbone of this portal. We're also expanding beyond wastewater and using some of the principles of what we've learned, especially in that environmental conditions and measurement side, to inform other microbiome and environmental samples, such as working with the Canadian Soil Observatory and other agricultural partners, which we're really excited with. But crucially, what I wanted to highlight is that this will stay a community-driven process with ongoing engagement to ensure that as the needs develop and as things change, we refine those fields and terms to make sure that it stays practical and useful to real-world surveillance needs. So in summary, the analysis is limited by the quality of our contextual data, and with this PHA4GE wastewater contextual data, we hope to use the best practices that we've developed to facilitate data sharing in a way that is modular and interoperable. It includes the relevant tooling and support material to make it easier for uptake engagement with the community. Hopefully that means it improves efficiency, and we know that time really is lives when it comes to public health, so being able to ensure that we can speed up that cleaning of contextual data. It streamlines data exchange, and hopefully it breaks down barriers. Now, I am literally on the last second, so I just want to say a huge thanks to everyone, but particularly the Data Structures Working Group, Dr. Griffiths, the wonderful Gillian Paul, who started this before me, and Rhiannon Cameron.
Thanks for that, Charlie. So now we have Eddie Lulamba, who's going to be coming to the stage to talk about his work trying to develop a malaria data standard, particularly within the context of African sequencing.
Hi everyone, my name is Eddie Lulamba. I'm from the University of the Western Cape at the South African National Bioinformatics Institute. I'm also affiliated with PHA4GE. So today I'll be presenting some of the progress toward advancing malaria genomic data standard in Africa. So before talking about developing the standard in Africa, I would like to present some of the background so that we can understand why it's important to develop the standard in Africa. So first we need to ask ourselves a question: why are sequence data standard necessary for infectious diseases? We all know that contextual data are key to understanding and interpreting sequence data. This is because many initiatives have their own standard to describe the data that they are sharing. Things like different levels of details, different structural organization, and it is important, especially during outbreak, to determine the critical information that need to be collected first. This is because during outbreak, there is often one opportunity to collect critical information. So we need to collect those critical information first, and the more optional one can follow after. So there is need for standardization to streamline data management and integration, and thus support more effective public health intervention by improving the understanding of the disease outbreak, the burden of the disease. So what about genomic data sharing in Africa? So in Africa, people have become reluctant to share their data due to legal limitations, losing oversight on the data that they are sharing, and also sometimes not having access to their own data. Also, the informed consent may not be sufficient, and the sensitivity of genomic data and associated metadata. And for long-term sustainability, so in many African settings, many African settings are facing challenges in maintaining infrastructure, funding, and trained personnel for sustained research and data stewardship. Regarding the malaria genomic standard, challenges include diverse, different data sharing needs. So data is often shared internally rather than globally through different platforms. And many projects operate in silos for some reasons, with little integration or coordination to ensure interoperability. Secondly, high malaria burden within limited resources. The existing standards are often developed by in high-income settings and may not align with regional priorities, terminology, and public health realities. So these standards often include extensive metadata fields that are impractical for low-resource environment, technical complexity. Their technical complexity and level of details is not yet feasible or contextually adapted for use in many African settings. As a result, in many African settings, no data is being collected for global sharing or secondary use. A minimum feasible set of critical metadata fields is urgently needed in order to move from collecting nothing to collecting something that is meaningful and scalable. As a summary, challenges in Africa originate from a combination of infrastructure limitations, lack of standardization awareness, policy barriers, and underinvestment in local capacity. Addressing these requires inclusive long-term effort to co-develop standards, build trust, and support regional leadership in data governance and sharing. To address this, the pathogen genomic data standardization in Africa was created, which is a collaboration between Africa CDC, PHA4GE, and the GeneAP network, which is a platform to a platform for inclusive, sustained development of infectious diseases, genomic data standard, while fostering trust and strengthening regional leadership in data governance and sharing. So under this initiative, the malaria community standard is being developed to support harmonized collection, integration, and sharing of genomic data across Africa and globally. So the stages include the development of the draft standard based on the scientific evidence and community consensus. So these are the summary of what it is. So we're presenting some of the scientific evidence that we use to develop the standard and what the standard is. This is how it looks like. So I'm not going to explain this. And then the second stage is the circulation of the draft for review and feedback from stakeholders, including researchers, public health experts, and end users. So we circulated it into the GeneAP network, and then it's being used in the Agari platform to develop the database for malaria sequence data. And then we iterated the refinement based on the feedback we received from these collaborators. The following stage is the implementation of validation and practicality testing through workshops and pilot studies to assess usability and effectiveness, which we conducted during our last pre-conference workshop. So what we did, we were validating because we did not build the draft based on the need assessment, so we needed to validate the field that we're proposing in terms of accuracy, completeness, and consistency within and across initiatives. And then we are busy compiling and analyzing the workshop data, identifying priority area for revisions, gathering feedback to improve alignment with field realities, and prioritize feasibility in low-income settings. So these are the next stage. Basically, we are going to perform the risk assessment and revision by a multidisciplinary teams, and then we are going to submit all the feedback and everything that we'll be receiving from all those groups for the approval by the governance groups, and then it's gonna be the official publication and dissemination of the draft standards, followed by ongoing monitoring. And we're hoping, in conclusion, we're hoping that the malaria community standards will provide a foundation for harmonized data sharing and surveillance across Africa. And because it is grounded in regional collaboration, it will support interoperability, strengthen data governance, and enhance public health response. So I would like to acknowledge all these organizations and institutions and all the collaborators and volunteers who volunteers to contribute, refining data, curating everything that we've been proposing. And then I'd like to thank the organizers of this conference. And thank you also for your devoted attention. Thank you.
Thank you very much, Eddie, for that great talk on the malaria context, community standard. Next, I would like to invite Dominique Anderson to stage, who's going to talk about her work developing a neonatal-specific metadata standard schema for the global genomic surveillance programs in neonatal sepsis. Dominique.
Hi, everyone. So I'm Dominique Anderson. I'm from the South African National Bioinformatics Institute, and I'm going to be speaking to you about a neonatal sepsis-specific metadata specification. So neonatal sepsis is actually a bloodstream disease which occurs in infants under 28 days old. It is the leading cause of death in neonates, and it can be divided into early onset, whereby the pathogen is transmitted from the mother to the child during childbirth, or late onset, whereby the pathogen is transmitted from the external environment to the newborn. In low-income settings in sub-Saharan Africa, for example, over 1 million neonates die per annum, and this is a death rate that is 11 times higher than in high-income settings. And in low-income settings, gram-negative bacteria are often associated with this burden, and they exhibit substantial AMR. So using genomic epidemiology to study bacterial isolates is important for the for optimizing preventative and therapeutic measures, particularly in vulnerable patient groups. However, the collection of the bacterial genomes by itself is not sufficient, and to be truly informative and accurate and complete metadata should be linked with genomic data. The structured data not only completes the data set, but it also facilitates data discovery and is key to the FAIR principles. And it is for this reason that we sought to develop a contextual metadata standard which can be applied to neonatal sepsis. So what was our approach to development? We first started with a literature search, which allowed us to actually catalog the information that researchers document consistently, allowing them to be strong candidates for addition into the schema. It also identifies whether there are existing standards which can help form the backbone of ours. And this is where the modularity of PHA4GE standards can actually be used to accelerate metadata schema development. Experts often know what details are critical but underreported, and engaging with the stakeholders not only reduces blind spots, but it provides valuable insights into the practical requirements and challenges, ensuring that the schema aligns with real-world surveillance needs. Their feedback was absolutely integral into identifying requirements and refining the design. And this engagement is also absolutely vital for community-based development of a standard. The engagement process actually involved several online meetings, and this was, continuous feedback loops were also established, and this allowed us to develop the standard iteratively. We also used ontology-based methodology so that we have interoperability with other standards that are developed the same way. So what are the core features of this schema? One is a comprehensive data dictionary. It is a user manual that clearly and explicitly standardizes terms and their meanings. The data dictionary groups terms into classes and maps where possible to ontological identifiers. It also provides guidance as well as examples, and it also provides definitions. It also then provides the type of data type that's associated with each field. In this particular example, we have core features that include identifiers, sample collection and processing, as well as contributor acknowledgements. Given the etiology of neonatal sepsis, we also include identifiers related to facility information. We also include additional fields related to sequencing, bioinformatics, and QC, as well as AMR. So while a metadata schema actually defines what data you collect, it often doesn't make the semantic intent explicit. So in addition to the data dictionary, we are actively building semantic concept maps, which are structured representations that show how the metadata elements are related to one another. The contextual metadata template is available in a Google Sheet as well as an Excel format. And these formats are used due to their familiarity and their low barrier of entry for those who capture data. The fields are separated into 4 distinct tabs to deal with the personas who might be able to capture those particular data. We separate terms into clinical, laboratory, genomics, and AMR templates. And there is also a pick list of reference data which forms the basis of data validation and can be easily modified to add more terms if required. We have also included identifiers which auto-populate across all the tabs to ensure appropriate data tracking. And where possible, we create dependent drop-down lists. So for example, in this template where we select a specific country in column H, it populates a specific selection of associated granular location data in column I. The laboratory template and the genomic template permits pathogen identification based on molecular and genomic analysis to allow for technique-based comparison. And as previously mentioned, the AMR template is actually based on the antibiogram profile from the NCBI. We have also included, don't know where that went. So future developments actually involve the prioritization of adaptations to the schema to incorporate stakeholder feedback and to address the dynamic requirements of neonatal genomics surveillance programs. Testing the schema actually involves a series of evaluations to confirm that it functions as intended and ensures that all components interact correctly and meet the defined requirements. The process also includes user testing to validate the schema's performance in real-world scenarios. And integration focuses on deploying the schema within existing surveillance systems. It is absolutely essential to ensure that the new schema works seamlessly with current data management practices. And this phase also addresses compatibility issues and facilitates smooth data transfer between platforms. So finally, I would actually like to call for community input to refine the schema. This is my selfish way of getting everyone to join in. So I want to do this in a collaborative manner because the more engagement we can actually get, we will allow for more diverse perspectives to be incorporated and enhance the relevance and usability of what we have developed thus far. So I would like to first thank everyone that I have engaged with in developing this standard, and I'd also like to thank the organizers for allowing me to present this talk, and to you, the audience, for your attention. Thank you.
Thank you, Dominique, for that excellent talk. So for the next talk, I would like to invite Colin Tanui up to stage, and he's going to talk about the work he's been doing with Africa CDC on a minimal cholera standard for low-resource settings. Dr. Tanui.
So thank you so much. My name is Colin Tanui. I work with the Africa CDC as the Principal Technical Officer for Pathogen Genomics, and I coordinate programs within the Africa PGI initiative. I'm going to talk about the fragmented so I'm going to talk about from fragmented to integration. That is the advanced cholera surveillance through metadata standards. So what we have seen before is how do we make sense of the data that we generate. And based on the project that we have been carrying on within the Africa PGI, that is the Africa CDC through the Africa PGI, we have seen that the data that we generate will not make sense unless we have got the metadata that we, it's associated metadata. So for the Africa PGI 2.2, which was launched early 2024, we aim to integrate the genomic surveillance for public health action. And this includes end-to-end from surveillance, laboratory data, all the public bioinformatics. And this really ensures that we have that utilized by the public health institutions. We have a number of priorities that is for expanded functional capacity, not only within the Africa CDC, but the member state, and then integration of the genomic surveillance within the priority for the priority use cases. And also, not only that we want to implement this integration, but also we want to ensure that we have that timely sharing of the data for the public health action. And this should be of high quality that can be easily to be interpreted. And also, as much as we want to sustain the genomic ecosystem, there is a need for the strengthening of the sustainability and coordination, and this can be through the use of strategic documents or the guidance documents that we usually assist member states to develop this and integrate with the National Public Health Institute. A number of projects that we are carrying out post-COVID, there's expansion on the use cases, and this include cholera that we have got the malaria molecular surveillance that is covering most of the countries across the continent. You can see that, for instance, malaria, we have 9 countries that are working on malaria, and for the AMR project, we have got around also 9 countries, and then for the DETECT project, that is being implemented across 12 AU member states. One thing in common in all this project is that there's generation of data. Generation of this data, it's not only doing sequencing, but we have to see the downstream of this data set if we can use it for, to inform public health action. And one project that I'd like to focus on is on the cholera genomics. So we have got one project that is the ColGen project, and I know that I'm presenting this one on behalf of the countries here who are seated here. I know some of them, they're here, but they could have been even the best people to present this because they did this work. So the main aim is for this project was to build capacity on the cholera surveillance, or how can we use cholera genomics to inform for surveillance purpose. And we wanted to see, do we see the cross-border transmission using the genomic data? What is the AMR pattern? Do we have co-circulating isolates within the continent? So these are some of the questions that we wanted to answer. And based on the initial results that we got here, we really got to answer some of these questions, including the transmission lineages. So if you see, for instance, T15, which is co-circulating within the southern part of Africa, it is only, we saw this one in Mozambique, Malawi, and Zambia. And when we had outbreak in Zambia, DRC team led by Dr. Placid was on high alert to ensure that if there is any outbreak at the southern part of DRC. And they did collect some samples, and they found that there were some samples, there were some some of the isolates were grouping with the T15 lineage from the same site. Then you see similar pattern we observed in Nigeria and Cameroon. So they share the same lineage, indicating that there is a clear indication that there is a cross-border transmission. But we could not tell the directionality of this. We don't know if it was coming from country A or country B, but we have seen that there's evidence that there is a cross-border transmission. And in terms of the co-circulating lineages, we also found that there are some multiple lineages that are circulating. You see the one for Mozambique, they have got a number of the lineages that were circulating over time. And there are some other countries, let's say, for example, also the southern part, we saw almost similar pattern. For us to get this, there is one thing that was, that is both, I just wanted to zoom this one, like the T15. For us to get further information or good information on this, we needed to have the metadata or the complete metadata for some of these, for the data. During this work, we found some challenges along the road. We could not get to this without getting that data. We had some challenges, including the data gaps that I'm going to highlight here. So we had fragmented data, you know, I'm just going to give an example. We could not use over around 250 genomes because it had incomplete metadata that can assist us to feed it in the analysis. And this is one of the big problem that we faced. And for us to better understand this, we really wanted to know what is this data set, what is this, what we need, and what is the metadata that we can use. So I don't want to repeat this because I know Dominique has said more on all this, explaining further on the metadata, but this is a data that can give information or descriptive information over given variable. And we have got 3 types, that is the contextual, we have got the structured and the administrative part of this data. In terms of the data pipeline, for us to have end-to-end, we have the sample processing, whereby we'll have the from when the sample is being collected and being processed to the lab. Then we have got the lab part when it goes to the lab for or the microbiology part of the processing. We have the bioinformatics part of it, and then this data is being fed to the analytical part of it, whereby it informs what we or the results, inform results. And another thing that I'd also mention here is the feature. Yeah, some of the features like the AMR. When we did a retrospective study, we found that most of the countries, they do not have this, they do not capture this. They do not capture this information. And why do we need this is because for us to get all these results, we have to get the harmonized data set. We had to ensure that for us to get to consistency, we have to adapt harmonized metadata standards across the 7 member states that they were participating into this project. Some of the metadata standards that we adopted was the use of harmonized protocol for all these countries, from sample, how it's being collected, processed, ensuring that we captured a minimal metadata standard for this. And one of them was the aspect of the sample ID, that who collect these. Most of the geographical locations was captured. We had the aspect of bioinformatics, sequencing and bioinformatics, that we ensured that it was captured. So in terms of how did we ensure that it was implemented, we build capacity through the collagen within these countries, and then we ensured that there was a collaboration with the TFCC, PHA4GE, to develop these metadata standards. So in my earlier, I mentioned that we don't work as individual, but we want that collaborative approach, ensuring that we work together and get input from the experts, especially from the GTFCC, that they had already developed some standards, but we wanted to ensure that it's what is captured through captured by most of the member states. And then the other thing that we did was to develop of harmonized, user-friendly data. That is a Google Sheet that had all the variables. I think also I would say that Dominique did present these tools. We also adapted the same approach for the cholera. What we did was we first reviewed the the existing metadata standards, the one for PHA4GE, the one for the GTFCC, and then work with the stakeholders, that's the community, the AU member states, to see what are some of the essential metadata field that we should be capture. And then also we mapped to control vocabularies. When we had a meeting to work on this, we found that the member state was saying the same thing, but they are using different terms. So we ensured that we adapt one term which was common across, and then if there is other terms which was more different but having the same meaning, we ensure that it is uniform. And for us to ensure that we, whoever is filling these metadata standards, they use SOP. You know, every field had its own SOP to ensure that you know what you are filling. And we have got two SOP, and we intend to translate these to all AU languages. As I said, we have the current form, the current version, that is current version 4, and we have ensured that we have captured the pathogen collection and the processing part. We have the host information, we have environmental information, and I could say this, when we were developing these metadata standards, there were a question of should we include the environmental information or just use the clinical information. But after deliberation, we found that this field should be there, but you choose exactly what you want. What are you working on during that time? So if you are working on environmental samples, you click environmental samples, and all variables or metadata for environmental will be there. So if you are working on the clinical, then you can click the clinical metadata field, and then you get the correct variables for that. And then we also look into the pathogen isolation and information, bioinformatics and QC, and sequencing. As I said, the current form we have got in Google Sheet format with accuration implemented, and as well as Excel format. And though we still working to see, because we're still working to see how we can put in the Excel format, because there is some challenges for some of the fields. And what we have found so far, we have around 80 fields, which include date, pick list, and the free text option. And as well as I said, we have got the two SOP being developed. And so far we have the field selector tool developed. We are already, this one has been developed. And the ongoing effort include the stakeholder engagement, because this is a living document. We'll still get improved as we go along. And then it will be translated into different languages, and then user testing. And we have put all this information in the Agari platform that as we are testing this one, we wanted to start with Agari first to ensure that this it can be used. So having said this, I want to say thank you and acknowledge the member states who really work hard on this field to ensure that we are where we are today. So thank you so much for listening.
Thank you very much for that talk, Collins. And so we now move on to the last talk of this session. Hina Salimuddin is going to come up and talk about her work on ClebNet the ClebNet metadata repository for really doing enhanced contextual data for Klebsiella pneumonia species genomic surveillance.
Hi everyone, my name is Hina and I'm a research fellow from Monash University from Melbourne, Australia. And today I'll be discussing the ClebNet metadata repository, a source of enhanced contextual metadata for genomes within the Klebsiella pneumoniae species complex. So Klebsiella pneumoniae is a bacterium that colonizes the human gut, usually without causing any harm, but it can spread to cause severe infections, particularly in hospital settings, where high rates of antimicrobial resistance are a problem. In fact, Klebsiella pneumoniae is the third leading cause of death associated with antimicrobial resistance. As this multi-drug resistant Klebsiella pneumoniae is being reported globally, the World Health Organization has listed it as a priority pathogen requiring enhanced surveillance and the development of new treatment strategies. This requires monitoring the spread and evolution of high-risk strains. To do this, we must collect genomic data from around the world to determine which types are spreading and how they're changing. There are already platforms that collect genomic data, including BigsDB, NCBI, ENA, and Pathogen Watch. Sorry, one second. However, what is often missing is the detailed contextual information known as metadata, which is essential for interpreting genomic data effectively. Metadata enables us to identify high-risk populations and the most common antigens, helping us to develop new vaccines. It can also inform the development of phage therapies and novel antimicrobials that are effective against resistant strains of the pathogen. Metadata has become widely recognized as a critical part of genomics research. The Genomic Standards Consortium was established after the realization that while large amounts of genomic sequence data were being collected, most lacked even the basic metadata information. The information about sample collection and laboratory methods were understood to be essential for interpreting results and conducting analyses. If we continue to lack metadata, then the genomics data used for surveillance, treatment, and understanding the pathogenic diversity will contain significant bias and uncertainty. For example, if you want to determine the prevalence of multi-drug resistance in a particular Klebsiella pneumoniae strain, you might retrieve relevant samples from NCBI. But there may be bias if only multi-drug resistant samples of that strain were sequenced, which would directly affect your prevalence estimate. Another research question may be whether a Klebsiella pneumoniae strain is a high-risk strain for bloodstream infection, but you won't be able to answer that if you cannot confirm that the data you have does come from blood samples. Overall, we need this metadata to design robust analyses to look for genetic associations because it allows us to consider the underlying sampling biases and confounders in the data. On NCBI, the total collection of prokaryotic genomes is over 2 million. For Klebsiella pneumoniae, the genomic data available on NCBI is still quite extensive, of 98,516 genomes. This has greatly enhanced our understanding of the biology of Klebsiella. However, we are still missing the metadata component. Information such as when and where the isolates were collected and under what conditions that they were collected is usually missing for these samples. Either the database does not collect this information or the database does ask for this information, but data contributors do not have that on them. So for Klebsiella pneumoniae, out of the 98,516 genomes, only 35% had data on the isolate country and isolate source information, and 37% had a recorded collection date. So to address this metadata gap, the ClebNet GSP team has developed a repository to store contextual metadata for all Klebsiella pneumoniae isolates. The database aims to collect enough contextual information to allow the future reuse of genomic data in research, while also not asking researchers to just send all the data that they have. The project includes a template for a guide page that lists each metadata field under one of 4 sections. The first section focuses on what the isolate is and when and where it was collected. The template explains which fields are required and what each field represents and how to complete them. Some metadata fields use predefined categories, making it easier for researchers to reuse this data in future research. And there are also open fields as well, which allow more detail to be provided. The isolate metadata section contains a total of 36 metadata fields, although not all are mandatory. Overall, it captures key information about the isolate's origin and associated host. The second section is for sampling information. This asks for details surrounding isolate collection. Things such as the purpose of sampling these isolates, the study population that they come from, whether the genome data was only sequenced due to the isolates showing a particular characteristic, such as antimicrobial resistance. This section will greatly help with reusing genomes in future research, as it allows you to select strains that have the least confounding factors. For example, let's say if you have a water surveillance study where you've collected isolates from fresh and wastewater, and these are routinely collected and stored. Let's say each year 100 ESBL-producing isolates are randomly sequenced. Because only ESBL-positive isolates are sequenced, their frequency in the environment can't be estimated, but you can see how ESBL genes overlap between different reservoirs, and that can be compared. In the template, you would report the sampling purpose as routine surveillance and the study population as fresh and wastewater. As the isolates were selected for sequencing only because they are ESBL producing, in the selected organism trait, you will input ESBL producers. This will provide detail of potential selection bias for future research. And since this is surveillance and it's ongoing, you would input the start date and leave the end date as blank. And overall, we think this approach provides a good snapshot of how, when, where, and why the sampling occurred based on the methodology and the study. So we have incorporated the principles of the PHA4GE Microbial Data Sharing Accord into this metadata repository and template, either in our guidelines or data fields. For example, we ask that only de-identified data be sent to us for human-associated isolates in accordance with Principle 4 of the Accord. We recognize that these standardized principles for the secondary use of data increases confidence in data sharing and for contributors, and provides adequate safeguards against data misuse when being reused in research. There are a few exceptions to this, given the nature of the project. At some point, we do want to link the metadata to external platforms, so those wishing to contribute data to the metadata repository would have to waive Clause 3, which requires consent every time the data is used. Clause 2 and 7, on the other hand, we have made optional and may be waived at the discretion of the data contributor. If contributors prefer that their data only be reused or included in publication after reasonable efforts have been made to contact them for collaboration or permission, they can indicate this in the relevant fields during their data submission. We think organizing the database around these key principles will encourage data sharing into the repository and remove any uncertainties contributors may have about their data being misused. We have a structured process for data submission and validation in line with the Accord as well. Contributors receive templates and submit de-identified data via a secure inbox. Submissions are stored securely. The submissions are stored securely and are validated for consistency and genomic linkage through an automated validation script, as well as some manual checks as well. And any resulting queries are resolved with the contributors. Finalized metadata is uploaded to the public metadata repository GitHub page. So this metadata project is one component of the broader goals of the CLEBNet Epidemiology Consortium. The consortium aims to collate publicly available Klebsiella pneumoniae species complex whole genome sequences alongside matched isolate source and sampling metadata through widespread global collaboration. We encourage researchers around the world to join the consortium and to contribute to the metadata repository. Those who join and submit metadata have the opportunity to be part of collaborative research projects and publications. All members can also actively participate in the analysis working groups and be included authors on those outputs as well. I've provided the links to the terms of reference and the registration form for this consortium at the end of the presentation for anyone interested in joining. And so far we have 77 people who've joined from 24 different countries, and we hope to see more participants from a broader range of geographical regions. But the response has been encouraging given that we've only invited people, it's been a couple of months since this project started. We currently have more than 5,000 records on the repository, but are looking to add more from the big genomic projects that already have data publicly available. So in conclusion, WHO has listed Klebsiella pneumoniae as a priority pathogen as it is becoming increasingly difficult to treat and is spreading rapidly. While we have genomic data, we are missing metadata that allows accurate interpretation of data and the reuse of these genomes in future studies. In response, the ClebNet GSP group has created a metadata repository with the aim of providing this contextual information and easily linking it to existing genomes on other platforms. The group has also established the ClebNet Epidemiology Consortium to facilitate a collaborative effort of curating this metadata and producing research outputs. I'd like to acknowledge the WISE Lab and ClebNet GSP team for their contribution in the development of this metadata specification and repository. I'd also like to acknowledge the ClebNet GSP Epidemiology Consortium members. We have already received metadata from a few members, so that's very encouraging. I've also attached links to the metadata and the template to the metadata template as well as the registration form for anyone who's interested in joining the consortium. Yeah, thank you.
Thank you very much for that talk, Hina. I would like to now invite all of the speakers up to the stage and open the floor for questions. So if you've got a question for any of the speakers, please put your hand up and someone will be roving around with a microphone to hand it to you and probably try and direct to all the speakers or a single speaker if you've got that specific question. So thanks, everyone. And if no one puts their hands up, I'm going to ask them awkward questions and we're all going to watch them squirm. So I will throw to the first question while people are gaining confidence. So, we've seen a lot of work that goes into developing these standards, right? A lot of resources. You're developing at the cost of all of your labor to do this work, the cost of getting people together to consolidate it, and the cost of doing testing. How do you, when you're so far upstream from the return on investment, how do you demonstrate it's cost effective to policymakers to do this? And I will open it to everyone.
I think we all know we're throwing it to Emma, right?
Well, I think we kind of touched on it this morning, right? Being able to have a business case, the before and after, right? To show you have messy contextual data. Here are the things that are incomplete metadata. Here are the things that you weren't able to do versus, the before and after. After, here are all the wonderful things that you can do. And I think that business case is strengthened by the reusability of the data, the sustainability, the longevity of the data. So being able to use data sets to answer a variety of different questions means you don't have to sequence as much, right? And that it all speaks to the return on investment. So you put in some money, you've got these powerful data sets that can be reused by yourself, but also can be reused by others because you've carefully documented the methods. You've provided the right kinds of information. It's in a structure that is machine readable and understandable by other users. So we really do need to be collecting those examples, those horror stories and those success stories to be able to feed those back to policymakers.
Okay, thank you very much. Any questions from the floor? Got two at the front down here, the roving microphones. I don't know where the roving microphones are.
Hi, Andrew Whitelaw from Stellenbosch University. I'm a clinical microbiologist, so probably one of the people who generates the organisms that feed into into the genomics and so on. So I have one specific question, and I think it's for Emma. I think you spoke about the QC issues in the metadata. Do you build in QC around the laboratory processes that go into generating the organism in the first place? So and specifically around some of the phenotypic AST data and how do you handle that? And then I'll throw my other question out, which is a more generic one, and I'm not sure if there's an easy answer for it. When you're developing metadata standards in general, what do you do about trying to sort of future-proof them and sort of building standards that sort of might be useful in 5 years' time, which is a bit difficult because we're not quite sure what's going to be available then, as opposed to kind of making it bare bones, let's keep it simple for what we need now. Thanks.
So I'll try to remember all the questions that you asked me. The first one about different kinds of QC. So right now the QC is really aimed for the sequence data. That's not to say that we can't have QC for the isolates. And actually, that was a really great question. I hadn't really thought about that before. Again, the standards that we developed are mainly aimed at the genomics part. But that's not to say that we can't be providing QC that was beneficial for biobanks and things like that. So, I mean, you should come and join the working group, and then you can help us make those standards. But maybe we can talk more about that after. I mean, we do try to capture elements of specimen processing that produce the isolates that make the genomics data. But I don't think it's going to be comprehensive enough for what you're talking about. But we should talk more about that. The second part was, how do we build standards that are like future-proofing for a long, to make the data useful for a long period of time. So, I mean, we try to talk to a lot of stakeholders. We try to gauge kind of where the field is going, but we don't have a crystal ball, right? Again, I think the modularity really helps. I think the standards that we developed, like the SARS-CoV-2 standard, was really built around amplicon sequencing, tiling amplicon sequencing. And as we started developing more standards, we knew that there were different things that people were doing, metagenomics, for example, and microbiomes. And what we can do is, that commitment to updating standards means that we can retroactively go back and add additional fields and terms or additional modules to our original data standards that make them, that increases their longevity. And we've done that actually before with wastewater getting into metagenomics. We had another One Health AMR standard that we developed for Canada. And there was a lot of interplay between what we were doing, developing the wastewater standard and updating the One Health AMR standard. So,
it ties into that sort of generic as it can be, but as specific as it needs to be, which is where the ontology is coming really handy. And as Emma alluded to, we developed these environmental condition and measurements module with these additional terms that came out of our needs assessment on the wastewater specification. But actually, most of them, whether it's water temperature or soil temperature, they ended up getting moved into this GRDI AMR package that we use. And so actually it really was, we picked all of those up and put them in, really with very little effort. So I think trying to make sure that you're not too specific. When we worked with ENA and NTPI to develop that PHA4GE wastewater attributes package, the reason we developed new terms was because a lot of the field names were wastewater specific. Now, if people wanted to look not just at wastewater, but other types of water, that wouldn't be able to apply. So we wanted to generalize that. So I think that's one of the other aspects that we've looked at and that we try to manage with ontologies.
So basically, I think standards are adaptable. So as new technologies come online, we can adapt standards that have come before it to bring it up to speed with new technologies. Hopefully that answers your question.
I feel like I might have to play translator.
I'm sorry. Sorry.
Any other questions?
So if I could add on what my colleagues have said here, is that there is the compulsory field that we usually ensure that this is captured. But as going forward, there are some of the optional field that it can be compulsory in other pathogens. For instance, if you, let's say, for example, you are looking for malaria and then you are developing for cholera, for instance. So it might not be compulsory for malaria, but it can be compulsory on the cholera part of it. So there is that need to capture all of them, but depending on the pathogen that you are looking at, then you capture the specific one, the compulsory one, but you still have the optional part of the metadata standards.
Hi, here.
Question in the corner there.
Andres Arandadias from IPSN Global Barcelona, malaria genomics. So my question is going to be for Eddie, but I'll try to generalize it. And going back to what Emma mentioned this morning on the need of policymakers to be in line with WHO guidelines, I was wondering how those guidelines for use cases where there's clear use and recommendations for how molecular and presumably genomics data is used and that's therapeutic efficacy studies and diagnostic resistance in malaria, how that is incorporated, what are those mechanisms, more like the top-down approach rather than the community approach. And I don't know if there are experiences in other pathogens for where there is guidelines from WHO or Africa CDC or any of those bodies, and what are the mechanisms to successfully incorporate that?
Okay, go ahead first. So I would say, so that's a great question. Thank you very much for that. I would say, well, so I think this is kind of what I was asking this morning about the need for maybe not primary contextual data standards, but aggregated standards. Because what we were learning at the conference was that a lot of times data sharing occurs in meetings where people are presenting trends of resistances in slides, right? Rather than they're actually sharing genomic sequences with metadata. And so I think the policies that are in place right now speak more to the aggregated data than they do necessarily to the individual data. But the short answer is that we are working through that right now. And maybe Eddie can speak to that more, but I would say that they are one of the stakeholders, a very important stakeholder, but we're looking into that right now.
Regarding the malaria standard, what we've done is a bit different. So we were trying to include many stakeholders as possible. That's the one good thing. So, and then we developed the standard based on the consensus coming from them on top of the scientific evidence. And then after that, so develop what we call our first draft, then we'll validate it, test, after that testing it, and then when everything will be done, we'll circulate it, gather all the information from all the stakeholders, and then we'll collect all those information, submit it to a group of experts to actually, based on those information, to work toward that. I hope it answers your question.
If I can add to that, so I've had experience of that with the cholera metadata specification, and what actually happens is we talk about the sort of community approach to sort of developments, but what you actually find when you're speaking to the community is the community eventually says to you, yeah, but there's this other standard that we want to use, so you need to align it with that standard, and then you've got to sort of make that part of your development, because people don't want to have to enter, say, case report forms twice, especially in sort of resource limited settings. So you've got to be able to incorporate that into your design, but strangely enough, it comes from the community development and not from the top down.
Any more questions from the floor? Got a hand from the front here.
So I've heard some really impressive stuff about kind of working upstream from your standards to the genomic databases. I mean, I had the great pleasure of helping with the Africa CDC data science training last week, and the number one comment from everybody was that how awful the incoming data was that they got to deal with. We know that a lot of that is coming from the fact that people are doing data entry twice, they're doing manual data entry, and then even when we're dealing with data platforms, then we have a outbreak of Rift Valley fever virus in West Africa at the moment, and then it needs to go into laboratory information systems. I'm very happy to hear about the data harmonizer tool, but what other opportunities are there to work with, for instance, laboratory information systems, data entry tools like ODK is one that I've heard about, other things to basically make it easier, maybe an R package that can take your specification and then help with validation. Where's the future for moving downstream towards capturing making it easier to capture clean data?
jump on that one, and then Emma will capture anything that I've forgotten. So we operationalize through the data harmonizer, so thanks for mentioning that again. But it is important to remember that the data standards and the tooling should be distinct. And although we operationalize through that, we actually use the linked modeling language schema as a way to do that. So actually you could take the frameworks which has the data standard, the validation, whether it's an integer, whether it's a float, whether it's a pick list, which we refer to as an enumeration, and port that into any different tool that you like. We use the data harmonizer because it's built in-house, but it could be anything. I mean, I know that tools like EpiCollect that was developed with Oxford University and Wellcome Trust Sanger have used JSON schemas to help with capturing the field, for instance, so being able to use the lat long from your phone and things like that to be able to speed things up. So it doesn't have to just be one tool, and I think that's something we need to consider with data standards, is making sure that we express it in a language that is machine readable and can be supported in lots of different types of tooling so that we can engage with the right communities and, exactly as you said, make it easier from the source of capture and streamline it from there.
And I mean, just to add to that, there are different ways to implement standards in data collection instruments. Sometimes it's just putting the standards into a spreadsheet. I think you were showing us that. Other times it's integrating into a lab information management system. Sometimes if you're creating a new database, there's opportunities to be integrating standards into database schemas. But if you already have an existing database, people are not going to be perhaps super excited about ripping those things up and starting anew, right? And so what you can be using is data standards as a mechanism of interchange between different systems. Again, people have come to us and described this problem. We've already got data structured in a certain way in our systems. How do we get it into your standard? And you can be using tools. There are tools like LexMapper that already exist that help you automate getting it into the right format from whatever messy data you have upstream. LLMs, we were talking about trying to incorporate those to help with those transformations. But if you're talking about how do we start collecting clean data from the start, I think really it is like what Charlie was saying, is taking these data standards, the specifications, and integrating them into a wide array of tools, and whatever those tools are that are most appropriate for your organization.
Take probably some quick final comments from Collins. You look like you're ready to go, but then we'll wrap up the session as we're running a little bit over.
Yes, so thank you so much, Peter. One of the things that we also ensure that there is a flow of information from when the sample is collected is the sample ID, whereby from the surveillance team, the same sample ID is being fed to the lab part. Initially, there was a tendency of losing that ID, and then the lab generate another ID, whereby it would be very hard for you to link the lab data to what it was collected. So with the metadata standards, for instance, the one for cholera, we ensure that the sample ID from the field is being captured in the lab, and it's not lost along the way, to ensure that there is a linkage between what is being submitted from the field and what is being analyzed in the lab. Yeah, I think, Dominique, do you want to add anything on that?
Oh, so just based on that, that is the same as the sort of neonatal sepsis one, where you sort of you make sure that the specimen ID is tracked across all different things. So if you have different tabs, for example, because you want to make sure that people who are knowledgeable in capturing that piece of information capture that information only, and you're not sort of bombarding them with this big, massive template that's 150 fields. Then you have to put things in place that make sure that there's efficient tracking across all of those tabs. So depending on what kind of tools you're utilizing, you have to put very smart measures in place to make sure that the users find it friendly enough, and that there's not a steep learning curve, because if there's a steep learning curve, people don't want to use it. They'll just go back to their normal way of doing things. So I think part of developing the metadata standards is not just developing these ontologies and these terms and these things. It's developing the tooling and the things that are going to make it as user-friendly as possible, and to facilitate the use of them. Because if nobody's using them, what's the point?
Thank you all very much. And please join me in thanking our speakers one more time. Sorry, we're unfortunately out of time for this session.